Much of your work concerns processes of biochemistry and molecular evolution as they bear on photosynthesis. I know you're working now, for instance, on the manipulation of the photosynthesis of algae as a potential source for renewable energy. But the reason I wanted to speak with you is that your team has also been doing some really interesting collaborative work in the humanities. In particular, you've been thinking about applying principles of evolutionary analysis and biogenetics to the history of literature, the bibliographical study of manuscripts, and so on. Can you talk a little bit about this work and where it's taken you in recent years?
Yes, of course. So one of the things that we're interested in from a biological point of view is trying to use the information in DNA to understand evolutionary relationships between different groups of organisms. And the idea behind that is that during evolution, mutations happen in DNA, and those mutations get propagated, and more mutations will be added to them. So that as species diverge in evolution, then more differences accumulate in the DNA. And one of the things that we do as biologists is use the differences, if you like, between the same piece of DNA or the equivalent piece of DNA in different species to work out the evolutionary relationship between those species. And it occurred to us a while ago that actually that process of copying with the incorporation of changes that then get propagated is actually very similar to what happens as texts are copied or were copied manually in the days before printing, with changes being made and those changes being propagated as those texts were copied in turn. So, and knowing that a lot of people are interested in working out how the extant versions of a text are related, we thought maybe we can apply our evolutionary biology techniques to try and work out the copying history of sets of texts, because it's actually exactly the same kind of problem.
So it's
sorry.
It's an analogy between what we in the study of text call stematology and what you in biology, I guess, call phylogenetics, or
Absolutely. So it's exactly the same process is happening in both cases. And stematologists have traditionally done all their kind of kept all their data in their head and worked out schematic trees very, very painstakingly. And we thought, well, maybe we can try using our computer programs to answer the same question, but much more quickly. And so we got together with Peter Robinson, who's worked for a long time on the Canterbury Tales, who actually was also very interested along similar lines, as it turned out, and so he gave us various data sets of his, and we tried putting them through our evolutionary tree building programs, and the result was very satisfying, because you can actually get very quickly a tree which is remarkably consistent with the ideas that textual scholars have had for a long time on how different extant witnesses to the tales are related. And then since then we've been applying the methods to quite a lot of other traditions as well. So we've done work on Dante's Monarchia, we've done some work on the Greek New Testament, lots more work on the Canterbury Tales, some work on the poems of Herrick, and lots of different data sets.
Are you finding a ready audience for this work? Is the work that you're publishing finding finding a readership?
Yes, it's interesting, because people seem to be quite polarized, actually, and there are those people who really like this approach and are adapting it and using it themselves, and there are other people, I won't name names, but there are other people who really don't like it at all, and argue that it's completely inappropriate because texts change in quite different ways from how sequences change. And yes, it's true, there are some differences, but actually there are a lot of similarities as well. And I think what's important is that we're not trying to argue that the computer is somehow giving you the right answer that you can't get otherwise, but what we're doing is giving people a tool, if you like, that allows them to look at a lot of data very quickly. So it's a kind of complementary to the kind of traditional established scholarship. It's certainly not a replacement for it.
Right. And another complementary method that I know that your lab has been involved in recent years is the analysis of DNA from parchment, the phylogenetic analysis of the actual material substance of medieval manuscripts. I know there's some North American scholars who've been doing that kind of work as well. Timothy Stinson comes to mind.
Sure. Yeah.
What does that involve, and what are some of the opportunities and challenges you see in that work?
So what we do there is, because parchment is basically just dried animal skin, it should have retained in it DNA from the organism, the animal that gave rise to that skin. So in principle, you should be able to extract some of that DNA from the parchment and find out, for example, what species of animal it was from. Now, sometimes if you've got a great big piece of parchment, then it's got to have come from a cow or something like that. But if you have something smaller and more delicate, maybe it's not always clear. So at a simple level, it can tell you what species it came from, and what we thought would be really interesting would be if you could actually take it further than that and get a kind of genetic fingerprint off a piece of parchment, so that you could perhaps compare it with parchments whose provenance you knew about, and therefore match them up. So you could say, well, this parchment whose provenance we don't know about came from a very closely related flock of animals to these parchments that we know were from a monastery in such and such a place at such and such a time. So that's, in my kind of wildest dreams, what I would love to do is to have a huge database that has all parchments with all known provenances in it, and every time you get a new interesting thing to look at, you can match its fingerprint up with that database. But that's a long way off at the moment.
I imagine archivists and librarians might have problems with the mechanism of sampling, taking samples.
Absolutely. Again, people vary quite widely. So my college in Cambridge is Corpus Christi College, which has the famous Parker Library, which has a stunning collection of manuscripts in them. They always look at me very suspiciously when I come into the library, worried that I'm going to start kind of cutting little bits out. But yeah, so it is a real problem that at the moment the techniques that we have are destructive. So you need a few square millimeters of parchment to extract the DNA from, and that's a real problem from the point of view of people. You can't do anything that's irreversible to a text, a parchment. So I think what we need to do is to try to develop non-destructive ways of getting DNA out of bits of parchment, so that there are fewer concerns like that. And of course, the hardcore among the conservators and so on will say that actually even by taking DNA out, even though you don't visibly change the structure of the parchment, by taking DNA out, you're changing it. So, maybe you shouldn't even do that. But I think you have to balance it against the information that you can get from all that data.
Right. Well, what both of these kind of constellations of research have in common is a different way of seeing the book than a humanist might. So I, as a literary scholar, I look at a book and I'll see words on the page. If I'm bibliographically inclined, I might do a physical description of the book, of the manuscript. But I guess one of the questions I'd love to hear you speculate on is, what is a book to a biologist? What does a biologist see when he looks at a book?
It probably depends which biologist you talk to, and I don't know. I think most biologists like a good read, just like anybody else. But I think what's quite interesting is that as biologists, we're used to thinking of the nucleus of a cell with all the DNA in it as this kind of big information source, because that contains all the information for making a cell. And maybe when you look at a book, you kind of think in a similar way that, here's a huge amount of information that's packed into a really small area. And there are a lot of interesting parallels between them, because in the DNA in the nucleus, the cell has to know how to access the right bit of information at the right time. And looking at a book, we have to be able to access the right bit of information in the book, as and when we want it. So there are quite a lot of similarities like that. And I think for me, it's been very satisfying to use these kind of genetic tools that we use, because when we're teaching students about DNA, we often use books and writing as an analogy for DNA. So to be doing it the other way around and actually using DNA as an analogy for books and writing, I think it has a kind of balance to it that's quite satisfying.
Are there other places where you see these kinds of collaborations between the laboratory sciences and the humanities going on? Are there other applications for the kind of phylogenetic analysis you're doing, for example?
It's certainly true that anything that follows this process of copying with the incorporation of changes that are inherited can be analysed by these kinds of phylogenetic tools. And so we've actually coined the term phylomimetics, because what we're actually doing is looking at what Dawkins would call memes and how memes evolve, and looking at evolutionary relationships between memes. So people have done work looking at the evolution of languages, looking at how different languages are related using phylogenetic techniques. And there's some very elegant work being done on those. People have done some very nice studies looking at cultural, or looking at artifacts. So, for example, Persian carpets. There's a beautiful study done by Jamie Terrani in Durham in the UK, looking at the designs on Persian, and what he's trying to do is to answer the question: are the designs and are the weaving techniques passed down vertically from parent to child, i.e. mother to daughter, or are they transferred kind of laterally, perhaps by merchants going from one village to the next, saying, can you make me one like they have in the village next door? So what Jamie did was to look at lots of different examples of carpets and encode the designs and the weaving techniques in a string of characters like a DNA sequence, and then use phylogenetics to look at those relationships and try to answer this question of whether it's kind of tree-like, mother to daughter, or whether it's a network, if you like, of communication between different villages. And by and large, it's quite a tree-like evolutionary pattern. So there's good evidence then that it's parent to child transmission. So you can answer very interesting anthropological questions using those kinds of techniques as well.
Well, thank you. This is a fascinating amalgam of work. I wish we could talk about it all day. But I do really thank you for joining me, Chris, and thank you for sharing your thoughts.
Great pleasure. Thank you.